We'll move to the next session presented by Keerthana. Topic is SERS and cancer, illuminating the path to early detection. I welcome Keerthana for the session.
Hi everyone, I'll present my slide now. Is the screen visible?
Yes, Keerthana.
Uh, Good evening everyone. I hope all of you had a great time with the webinar going on. With me being the last session, I know I'm standing between a much needed break and you. So I'll try to keep it short and interactive. So let's. I hope everyone gets interactive with the session, and I hope everyone likes this session. So my topic is SERS and cancer, illuminating the path to early detection. So a small introduction about myself. I am Keerthana Nagarajan. I am a scientific content writer in Scioverleaf from May 2024. I am also a final year graduate in Bachelor of Science in Microbiology, studying in Ethiraj College, Chennai. So, what do you know about cancer? Can anyone answer? Would you guys know about cancer? What is your thought about cancer? Okay, so cancer is the abnormal and uncontrolled cell growth, which is it causes a group of disease. So there are three types of cancer: carcinoma, sarcoma, and leukemia. So carcinoma is the type of cancer which
Sorry, Keerthana, for
causes,
interrupting.
yeah.
There is a background noise. Can you please check that?
Yes, 1 minute. Yes. Now,
Yes,
so
yes,
sorry
go
for
ahead.
that. Sorry for the inconvenience. Let's continue. So what we know that cancer is the abnormal and uncontrolled cell growth which causes a group of diseases in different places. So based on the places where cancer is caused, there are three types of cancer: carcinoma, sarcoma, and leukemia. So carcinoma is the type of cancer which is caused in the epithelial cell tissues. So your cervical cancer, breast cancer are a type of carcinoma cancer. Sarcoma cancer is the type of cancer where your connective tissues, muscle tissues, adipose tissues are affected. So sarcoma is the type of cancer which causes bone cancer and etc. Leukemia is the type of cancer where your white blood corpuscles are affected. Uh, there are multiple carcinogens type of causes of cancer, and the five of them are listed here: oncogenesis, tumor promoters, tumor viruses like hepatitis C virus, human papilloma virus, chemical carcinogens like ethidium bromide, SDS, radiations like MRI, X-rays, and all. So there are uh, traditional diagnostic techniques which have been used up till date for diagnosing cancer. So, physical examination, imaging techniques like X-ray and CT scans, uh, biopsy. There are two types of biopsy: needle biopsy. Needle biopsy is the one which I have represented here. Endoscopy biopsy is the type of biopsy where we insert a camera and take out the sample. Blood test, also known as liquid biopsy. Endoscopy is where we insert the camera in the body using a flexible tube and visualize the organs. We do not take the sample, which is done in endoscopy biopsy. So, cytology or pap smear. So, cytology or pap smear is the type of diagnostic technique where we use a microscope to analyze the cancer tissues present. So, biomarkers. Biomarkers are the characteristic indicator of normal biological processes. So, when something goes wrong, we have this acts as an indicator. If something is normal, this acts as an indicator. If the biomarker level is more than the required, or if it is more than the required standard, or if it is just present, its mere presence could be an indicator of something wrong with our body. So cancer biomarkers are present in tumor tissues, as we all know, but they are also present in serum, urine, saliva, sweat, and many more excretions from our body. So there are several types of cancer biomarkers: long non-coding RNAs, mRNAs, microRNAs, DNA, proteins, cellular metabolites, exosomes, and organic materials. So, an ideal biomarker should be sensitive and specific. So, what do we mean by sensitive? Is that we should be able to uh, analyze the biomarker present in the sample. They should be sensitive to the tests. And specific, they should, for example, breast cancer, BRCA1 or BRCA2 is specific for biomarker, cancer biomarker is specific for breast cancer. So they should be specific for what cancer we are denoting. So here we come to surface-enhanced Raman spectroscopy, SERS. So surface-enhanced Raman spectroscopy was first discovered by Fleischmann and his team in 1974. But it is primarily based on Raman spectroscopy, which we all know is discovered by C.V. Raman. So, Raman spectroscopy is a powerful analytical technique that uses the scattering of light uh, to study molecular vibrations and chemical composition. When light interacts with the molecules, there is a scattering at different wavelengths, scattering of the molecules at different wavelengths, and with those wavelengths, we analyze the structure of the molecule and the properties of the molecule, because each substrate produces different types of spectral fingerprint. Spectral fingerprint is unique, like just like human's fingerprint, the spectral fingerprint is also unique for every molecule. So in surface enhanced Raman spectroscopy, what happens is that incident light is emitted, and we have a substrate, we have a metal nanostructure on top of the substrate. So this metal nanostructure is enhances the SERS signals. That is, they enhance the scattering of lights, which gives us more emitted lights. So why do we use SERS for cancer biomarkers? Is that we need accurate and complete diagnosis for cancer care. That which this is very important. Accurate and precise diagnosis is very important for treating cancer. So if we detect earlier, the preventing cancer would be easier, and treating it would also be easier. So this there are certain working constraints in traditional diagnostic techniques, where that is, we can get false positive results or false negative results, so they are not very reliable. So we use molecular tests which are reliable, and they are rapid and target specific. So this also is used because used for trace detection. Though biomarkers are present in very trace amounts, we can still use SERS. Again, coming back to the slide, where we see that this SERS, surface enhanced Raman spectroscopy, we use gold coated texture, where this enhances the substrate. We even though we have trace amounts, we can use this method to enhance the results. So this usually contains gold nanostructures, but we could use silver and copper too. Though if asked if they are reliable, no, they are not reliable. Usually, gold and plasmonic nanostructures are less. Gold and silver are more reliable than copper, because copper is gets affected by colloidal activity, while gold and silver are stable in the colloidal activity. So they are chemically specific. They are we can use cancer biomarkers to specify the type of chemicals, type of cancer we are using, and this primarily depends on the surface graded agent. So whatever surface we use here, whether it's gold, silver, or copper, uh, it primarily depends on the surface we use. So there are two types of detection by SERS methodology: direct detection, label free, and indirect detection, where we use tags and probes. I'll come to that later. But firstly, direct detection by label free SERS methodology. So we do not use labels or probes here. We do not use tags. It is completely based on the intrinsic properties of the molecules, and it was used to identify CTCs. CTCs are circulating tumor cells, which I'll come later. Indirect detection is the type of SERS methodology, where we use protective shell made up of metal nanoparticles, and these enhance the SERS signal. They also make sure that there is no cross interaction between the molecules. They are target specific and bind only to the specific molecules which are required. They also provide a protective shell which enhances the colloidal activity and do not get disrupted by the colloidal activity. So biomarkers in non-invasive sample. Non-invasive samples are the type of samples which are taken out of a human body without disrupting the physiological activities. So your sputum, saliva, sweat, urine are all types of non-invasive sample. So in sputum sample is generally used to determine lung cancer. So when we say sputum, most of the people confuse it with saliva. Sputum and saliva are completely different. Sputum cannot be produced by everyone. So that's a major drawback with sputum analysis. So what happens in sputum analysis is that we can identify three types of biomarkers here: NAP, EGFR, and CK. NAP is napsin, CK is cytokeratin, and EGFR is epidermal growth factor receptor. So when we use this method in SERS, what we do is we take the sample, sputum sample, and conjugate it with anti-NAP, anti-EGFR, and anti-CK, and they are run by the SERS molecules, SERS analyzer, and we get the when we analyze it in the SERS, we realize that there are the anti EGFR, anti NAP all conjugate with the NAP and EGFR and CK, and make sure that they are visible. So urine analysis, what happens is that we urine sample usually contains ERG, PCA3, and KLK2. So these three are generally the biomarkers of prostate cancer. So urine sample generally indicates the prostate cancer. Indicates prostate cancer. So what we do is, when we amplify these trace amounts of biomarkers using RT-PCR, and we add the silver nanoparticles, which will accumulate when there is the presence of these biomarkers. So when these accumulate, we can identify that the SERS has, by using the SERS, we can identify that this person is suffering from prostate cancer. So these are the types of this is a table which denotes the type of cancer for the type of sample type. So in sample type sweat, we do not use sweat a lot because sweat is not more reliable. We only get it in very trace amounts, and even though there is a cancer marker like fluorouracil drug, it is very rare. So if a fluorouracil drug is present in the SERS, it denotes that the colon cancer is present, and the nanoparticle we use is silver colloid. So these are the this table denotes the types of sample, denoting types of cancer. So next we come to biomarkers in invasive samples. So invasive sample is the type of sample where uh, the blood is taken out. Liquid biopsy or tissue sample is invasive sample. We are not getting into tissue sample because the main ultimate point is to reduce the stress given to the patient. So we will only be using liquid biopsy, that is blood tests. So by blood test we collect we can determine two types of cancer biomarkers: circulating tumor cells and extracellular vesicles. So in circulating tumor cells are CTCs are the type of cells which help in metastasizing the cancer. That is, we have a primary cancer and the cancer has to spread to the other part of the body. So what the cancer does is it releases circulating tumor cells into the blood, and it gets into an other part of the body and again starts another tumor. So this is done by circulating tumor cells. So we in this test what we do is we collect the blood and we incubate it with nanoprobe. So nanoprobe is nothing but the gold or silver conjugate which we will be using along with the uh, antibody conjugate. So after incubation we will centrifuge it, and during centrifugation we will use a filter paper where the unbound cells settle down and the bound cells remain in the top. So these bound cells are, when examined, are seen to be forming CTC sandwich complex, which is nothing but nanoprobe, that is gold nanoparticle or silver nanoparticle binding with the CTC cell. And when we analyze it, the spectral analyzer we interpret that the person is suffering from cancer. Extracellular vesicles. So extracellular vesicles are the type of cells which are used in communicating with other cells. Like these cells are very important in cancer because one cell when one cell is metastasized, the other cell should also know that it has to metastasize. That's what cancer is all about. Neighboring cells keep get affecting and the cancer spreads. So this is a very important feature in cancer, and when we determine that there is extracellular vesicles, it means that the patient is suffering from cancer. So in this experiment what they have done is they have collected melanoma cell derived extracellular vesicles. So the target EVs, extracellular vesicles, bind with the nano target molecules, and they form a complex which is again filtered by a filter paper, and they which is again filtered by a filter paper, and they form complexes. this filter paper separates the complex formed and the EVs and unbound EVs and unbound labels which are used. So this is again used to in a SERS, and the mapping is done, and we interpret that when the EV is elevated, we interpret that this person is suffering from cancer. So though there are a lot of pros of SERS, there are certain limitations. So the limitations are complex sample matrix. That is, they have to form the samples, such as we need to find the biomarker. If the biomarkers are not present in the sample by any chance, and if the person is actually suffering from cancer, then that would be a problem. We will not be able to diagnose the person with cancer. And reproducibility of the SERS signals. So the reproducibility issues is that signals can be challenging due to variability in the preparation of SERS substrates. So small change in the substrate structure, size, or distribution can cause significant variations in the result. Interference and cross reactivity is generally the overlapping signals from other molecules in the sample, which can interfere with the detection of specific biomarkers. And substrate specificity or substrate stability. SERS substrates may degrade over time. They are not very environmental friendly. If there are unfavorable conditions, they can degrade easily with not not they if the it does not have optimal temperature or humidity or exposure to reactive reactive molecules. They affect the performance. So I would like to conclude by saying that SERS is a powerful tool with immense potential in cancer biomarker detection. It offers enhanced sensitivity and specificity, though there are certain challenges which remain. With ongoing advancements in nanotechnology and computational analysis, we can pave way for better adaptation to the SERS diagnosis. Thank you.